you all may have heard in the media, there's a lot of great work being done on the human microbiome nowadays, but there's also a lot of misinformation out there too. And that's why I think it's so important to have people who are actually doing the work come talk to the public about it. So to tell you about and also to have our artist in the kitchen come talk to us too, because that's another very important part as well. So little bios about everyone. Here to the far side of the room is Owen White. He's the Associate Director of Bioinformatics at the University of Maryland School of Medicine and the Institute for Genome Science. And he's a prominent scientist in the Human Microbiome Project Central Data Repository. He was recently awarded the Benjamin Franklin Award for Open Access in the Life Sciences, and when he's not wrangling terabytes of sequencing data from thousands of projects, he makes very impressive metal objects with a giant laser cutter in his basement. So think about that part too. Because that's one of the neat emerging things, and something that really hit me when I started at IGS. I guess I should introduce myself as well. I'm sorry. I'm Ann Estes.
No, I didn't do it either.
No. But I'm a postdoc at the Institute for Genome Sciences, where I work on the dung beetle microbiome. And I'm also a science communicator as well, so I have a website called Mostly Microbes. So it's trying to talk about the human microbiome and microbes in general, and that, yes, there are pathogens out there, but there's also some that don't do so much harm, and those that are also helpful. But that was one thing that impressed me at IGS when I first started, was how many of the scientists actually are artists as well. So Emmanuel is actually, I guess I should go down in the order that you're in and not manipulate you. So Noel Mueller is our next person. He's our assistant professor in the Department of Epidemiology at Johns Hopkins Bloomberg School of Public Health. He's interested in using randomized clinical trials to unravel the interaction between diet, microbiome, and medicines used to control obesity, type 2 diabetes, and cardiovascular diseases. He also recently trekked through the rainforest in mountains of Venezuela to study the indigenous South American Yanomami tribe. Then there is Dr. Nancy Reagan. She is an associate professor at the Department of Organismal Systems and Adult Health at the University of Maryland School of Nursing. Her training originally was in nursing and midwifery in the UK, and was an advanced practice nurse primarily in labor and delivery for 19 years before working on her PhD. So she has a really awesome clinical aspect that she brings to her work, her research. She's interested in how birth practice influences the vaginal microbiome and preterm childbirth. And one of her long-term goals is to do research on maternal mortality in the country of Malawi. Is that, I'm saying that right? In South Africa, where she was born.
Black Africa.
I'm sorry?
Black Africa. Malawi's its own country.
Okay. Sorry. We have, and you had a nickname too. I was going to let you tell everyone your nickname when you were a kid.
Oh, yeah.
That was a cute story.
Well, I got sent to boarding school in England, and I grew up on a huge farm in Africa. And so when I went to England, they had no idea who I was. I really had no idea who they were. So they called me Mowgli from, the jungle.
Oh, okay.
Oh, that's great. Emmanuel, please say your last name. I messed up every time.
My way or your way?
Both ways.
Mugada.
Thank you. Is an assistant professor at the Department of Microbiology and Immunology at the University of Maryland School of Medicine, also the Institute for Genome Sciences. He's formerly trained as a physiologist and focuses on the application of microbial genomics, comparative genomics, and metagenomic approaches to the study of post-bacterial interactions and the microbial populations colonizing the human body. So he likes looking at lung bacteria, nostrils, and mouth due to conditions such as tobacco use, appendicitis, HIV, and MRSA. Then we have our two artists in of the crock pot. Can I call you that? So this is Megan and Shane Carpenter of Experiments. And their business is in Towson, which I highly recommend. It's
in Baltimore.
It's in Baltimore? Oh, I'm sorry.
Baltimore City.
Oh, okay.
Yeah, it's right on the border, but
It's right on the border. Okay.
proudly in the city.
Okay. They're proudly in the city of
And
Baltimore.
paying
Yeah.
city taxes.
And paying
Yes.
city taxes.
Oh, right.
There
That's
you
In
proud.
two places.
They use the traditional method of fermentation to preserve and transform local organic produce into nourishing foods unique sauerkrauts, kimchi, seasonal ferments, and kombucha tea. They began fermenting in an effort to dovetail their love of organic gardening and food as medicine. Now, as a business, I see the relationship between farmers and consumers as one of utmost importance towards rebuilding food sheds. The creations they make are nutrient-dense, probiotic-rich foods that are artful and delicious. They're inspired by both tradition and intuition. And the name comes from Hexology folklore that uses signs and symbols to protect relationships, secure harvest, and ward off the unwelcome. So, similar to that, their foods protect, support, and shield our immune systems, digestion, and overall well-being. And they've been featured in quite a number of publications, including Baltimore Style, The Sun, Local Palette Magazine, PBS, and named City Paper's Best Local Foodie Product. They've also taught a lot of workshops, which I've gotten to attend, at area colleges, farming conferences, and to local chefs. So they're provide a neat aspect of this as well. So I was hoping that everyone could give a 5-minute kind of description of your work and your goals and questions, and then how it pertains to medicine and microbiome. So
I think we should start on that.
you still have goals with your work as well. But Owen, how do start? And we do have markers available to you if anyone decides to schedule.
If I were just to describe what I do, I consider myself very much an engineer that help other people get work done. So when you do microbiome analysis, there's an enormous amount of sequence data that gets generated, much more than people are sort of able to analyze in kind of a one simple kind of desktop computer. So for the last 7 years or so, I've been funded by the NIH to help collect up data from other people who are generating it. So really there's good examples here of people who are sort of have an interest in a specific disease. And then we host it at our site. We do a lot of cleanup and refinement and often assist other centers with putting it into other public repositories or storing it and making it available at our site. So I mostly have a kind of a computational background. I'm much more interested in, ease of use for the scientific investigator out there to get their job done.
I actually come to the microbiome space from epidemiology. So I'm an epidemiologist focused mostly in the prevention of chronic diseases. And we spend a lot of time in epidemiology not just tracking Zika virus and other epidemics, but thinking about causality in humans. And so that takes the kind of constellation of animal research and human research and good design of studies, making sure that you're controlling for things like confounders that are out there. So kind of my interest is in the early life, the early origins of adult disease, and that is kind of how I came to the microbiome research that I'm doing today. So when I was doing my postdoc, we were looking at intrauterine factors that affect the programming of different organs in the human body that might affect diabetes risk and obesity. And we were looking at antibiotic use in pregnancy, we were looking at infections, we were looking at delivery mode and finding that they had pretty strong effects for the risk of childhood obesity. So I was at Columbia University. I went across town to collaborate with Marty Glazer, who does a lot of work in antibiotics, Maria Gloria Dominguez-Bello, who does a lot of work on C-section deliveries and how they alter the natural progression of the microbiome in babies. And we kind of joined up and did some epidemiology research looking at how these determinants of the microbiome affect human health, and then filled in some of the mechanistic gaps by sequencing things that the people who were doing the sequencing, collecting samples from babies and then sequencing and finding some differences between C-section delivered babies and vaginally delivered babies. And it's still a work in progress to kind of figure out how these differences in the microbiomes of the C-section vaginally delivered babies might alter the metabolism, but it's one example of things that people are working on in this space. But it also has the limitation that some of these differences might be due to confounding, because we know that C-section delivered babies and their moms are very different than vaginally delivered babies and their moms. So, thinking critically about this and getting to causality, got me into the space of trying to design rigorous clinical trials that we can randomize humans to either receive, for example, in a C-section, receive a seeding swab at birth versus not. If it's an emergency C-section, those moms are now becoming more interested in doing this, but to find out whether there's health risk or health benefits, Maria Gloria and I are trying to get funded a clinical trial to look at that. And then in adults, in terms of metabolism, we're looking at some drugs that might have gut effects. So we're looking at metformin, and we're looking at weight loss, because there's been a ton of studies that have looked at differences between obese and non-obese individuals and find enormous differences in composition, diversity, but obesity is a multifactorial disease, and every way that you get there may have its own effect on the microbiome composition, diversity, and each of those effects may be driving the differences that you see between different studies. So conducting a clinical trial where you isolate one of the ways of either getting to obesity or reducing body weight can help kind of determine some of those different pathways, the microbes that might be of interest. So further down the line, we might be able to hone in on those microbes and improve metabolic health. So those are kind of just some of the areas that I'm working in.
I've got some specimens for you. So I'm a nurse, and I'm an accidental transplant to the world of the microbiome. Nurses are we learn to be very astute observers, and we observe patterns. And so I can remember when I was doing midwifery, thinking about children that have necrotizing enterocolitis, and noticing that it was always the kids that we would give formula to that would get it. And other patterns, women, in England when I was training, we were under strict rules about how frequently you could do vaginal examinations during labor. Even then, some people would do more than others, and we would notice a much higher infection and postpartum hemorrhage rate amongst those women. So I sort of carried this with me through my life. And then my husband was transferred down to the Midwest, and I actually had just been foolish enough to go into medical school and absolutely hated it, and had no way to tell him that I really hated medicine. and I wanted to get out of it. And so I made the sacrifice of giving up, fortunately, and I was pre-docked into a program in Minnesota. So I went into the PhD program, and suddenly all of this observation, became, it really went to good use. So I started doing work on cesarean section and looking at how women, how did the section rate get to where it is, and carried on along that way. And then the NIH published an RFA for a grant, so I already had funding at that point. And they wanted us to look at preterm birth and diet. And at that point, somebody said to me, well, you should really meet Jacques Ravel at IGS. And so we got together and we put together a grant which is now funded by the NIH, and now looking at preterm birth. And every day is absolutely incredible, because it's like being in your first day at school. Because I am a clinician at heart, the microbiome has allowed me to connect differences that I never had a way of answering before. And so for me, it is like the missing link. I can look at things now, like the vaginal exam. I have a PhD student right now who's looking at that, and we've just got the data and looking at the number of vaginal exams during labor, and these fundamental, massive shifts in the composition, the diversity, and the structure, and now being able to understand, okay, we're also looking at inflammatory markers, so we can understand why the postpartum hemorrhage rate is higher, for example. So my work still remains very clinical, but every day is a learning experience. Every time I get all this omic data dumped into my lap, I have to, hit the books really hard for many weeks so that I can make sense of them, or come places here and pick their brains to figure out what we're doing. But it's really wonderful, and this is, I think, the most important thing for childbirth. And I don't know how we get that message out to the public, but it's really important that we do.
All right, so where do I start? So I did my PhD looking at bacterial adherence to the airway epithelium. So that was some time ago. And at that time, what we wanted to look at is how do bacteria successfully colonize the lungs, okay? So we know that in such cases, bacteria have to attach to a surface. That's the very first step. And then once they're there, then they will, depending on the circumstances, go deeper in the tissue and then create all sorts of issues. And so I studied that for a certain number of years, just looking at one specific bacteria versus one cell type, and it was all nice and neat because, you see the bacteria and they're all the same because it's all the same bacteria. And then the cute cells, and you try to see how they attach and what the bacteria do and what the cell response is, and et cetera, et cetera. And then at some point, the microbiome field started to catch up. So these were the early days, and when I say early days, that was like, not even 10 years ago. And then I started to realize that everything that I done during my PhD was basically complete bogus, because we know that in real life, there's never one bacteria completely isolated in the system. So at that point, I was doing something different, looking at antibiotic resistance in bacteria. And then I was like, well, wait a second, let me, go back to my PhD work, and maybe, we can revisit that, because we know that in real life, again, it's not just one bacteria, one cell type, but it's many different bacteria that are interacting with each other, and underneath that you have us, the host, and it's not just one cell type, right? So we have, the epithelium, we have the immune system, and everybody is either happy together or fighting everybody else. So the bacteria are gonna, it's war, right? So there's alliances that are done. Bacteria that hate each other, so that will fight against each other, and the immune system is going to come in and say, well, I like you, but you I don't. And so the system is a lot more complex than what I had done, 14-ish years ago for my PhD. So that's when I started to get into the microbiome and trying to really study a system that's a lot more dynamic and a lot more complex than what I had originally studied. So some application of that are what I'm currently doing, looking at what we call nasal colonization. So we are all colonized by bacteria, and approximately, I think, 40, 50, 60% of us are colonized by this bacterium that people call MRSA, so Staphylococcus aureus. And sometimes it's really bad, and most of the time it doesn't create any issues, but we know it's there all the time. The problem is that we don't know why it's there, okay? And for some people it's there, it sticks, people will have it for life, and other people, they get it, they lose it, they get it back, they lose it again. Some people have it and then they go to the hospital and get what we call decolonized, so meaning that there's an antibiotic that's applied, lose it for a little while, and then it gets back in there. But because there was an antibiotic, we want to know why and how it comes back, and if it comes back, in the same setup as what it was before. And the thing is also that there's also the bacteria in play. So we know that there are other bacteria, for example, that can inhibit MRSA colonization. So what we're trying to understand is how the system works and what the dynamics are. And the goal is, from a practical standpoint, so for example, when someone goes in surgery and gets what we call SSI, an infection at the site of surgery, most of the time people get infected by the bacteria they carry, right? So one possibility would be, well, if we know that people going on the surgery will get infected by the MRSA that they have in the nose, is, completely remove the MRSA in their nose. So that's what people have been doing. The problem is that when we do that, we remove a problem and replace it with something else, because obviously our nose is never sterile. So if you remove MRSA from the nose, something else is going to take its place. And sometimes that something else can be better, but sometimes that something else can be much worse. So part of the microbiome studies that we're looking into right now is what happens when someone gets decolonized, and because that antibiotic cannot be applied forever, what happens after? So do people regain these bacteria, or do they gain other bacteria, and is it a better or worse situation? Okay, so that's one project. So my projects are a bit more eclectic than what you've heard so far. So I've had a few different things. So that's one aspect. As Anne mentioned, you mentioned the tobacco, I think, in your. So I have that other cool project where we're looking at the tobacco microbiome. So it's not directly the cavity of people using tobacco product, although that's something that we're looking at. But in this case, it's looking at the bacteria on the tobacco itself. So tobacco, whether it's cigarettes, smokeless tobacco, you name it, is like cheese. It's a living product, okay? And it's not because you smoke a cigarette that you're burning all the bacteria from that cigarette. So, everything gets burned, at the burning site. But obviously, when the smoker inhales, those particles will carry bacteria inside the oral cavity. What people have realized is that in some cases, tobacco companies can do gamma radiation on their tobacco. And then we know that the all issues that the users will have will be different than when the tobacco is not radiated. The problem with that, so our goal is not to make tobacco, better for everybody else. The problem is that the bacterial content in tobacco product is not regulated. So we have a project that's funded by FDA, USDA to look into that and see if there are regulations that could be put in place. Because, I don't smoke, but so I had to learn about tobacco, and it's unbelievable. But there's no, sell by date. There's no, you buy a pack of cigarette, you don't know if it was produced a week ago, a month ago, or a year ago. But the type of experiment that we are doing is if you keep a pack of cigarettes, for example, in your pocket for a week, what happened to the bacteria on the surface of the cigarettes? And then if you use those cigarettes after a week, then do you get exposed to different bacteria? The key here is that the bacteria that we find on the surface of the tobacco come from the plant, but also the processing facility. And these bacteria are non-potential human pathogens. So the bottom line is that, so people get colonized by these bacteria from the surface of the tobacco, and we have no idea how and what these bacteria might be doing. We know that some of the carcinogens in cigarettes are actually the product of bacterial activity. So these bacteria are on the surface of the tobacco, but they're not doing anything. So they're metabolizing stuff and producing molecules that will be, bad for your health in addition to what was in the tobacco. So the goal here, again, is not to produce safer cigarettes, but to put regulations in place so that the bacterial content will be regulated the same way that we are regulating the chemical content of the cigarettes. And a third project, which is really, really, really cool, is to look at the impact of the microbiome in a transplant outcome. So the hypothesis here is that you can take, so in this case we're looking at kidney transplantees. And in kidney transplantation, the outcome of a kidney transplant after 5 years has not changed over the past 25 years, meaning that the rate of rejection has been the same for the past 20, 25 years. We have much better rejection drugs. We can match transplant and transplantees as best as we can. We haven't been able to figure out how to improve mid and long-term outcome for kidney transplant. So what we think is happening is that there's one thing that we don't control is the gut microbiome. So what happens when someone receives an organ transplant? So first, their metabolism changes because they go from not having functional kidneys and having dialysis to suddenly having a system that starts functioning normally. So it's a major shift in their metabolism. The second thing is that because it's a massive surgery, they get a lot of antibiotics that just flushes all the bacteria. So they will get some bacteria back, but because of, so they have sometimes, more antibiotics after that to avoid, post-op infections. They will have the anti-rejection. So, just put bluntly, their microbiome is pretty messed up. What we know is that in some cases there's what we call a chronic inflammation, meaning that the immune system has a low level activity that shouldn't be there. And what we have been looking at is what it does to the organ itself. What we have been showing, and actually the data is beautiful, is that the gut microbiome can modulate that, meaning that if you take different people and you change their gut microbiome, you can make their immune system attack, if you want, the organ faster or less, meaning that people would reject very quickly or they would have a delayed rejection just by playing with their gut microbiome. So what we are studying is to find ways to kind of like shut down that inflammatory reaction post-transplantation, not necessarily by playing on the microbiome, but the microbiome is just a vessel. So in this case, we could design drugs, for example, that we target the same pathway. So that doesn't mean that we will give yogurts to everybody post-transplantation, but the microbiome for us is just to see if we modulate the immune system through what happens in the gut, then can we have an impact on the outcome? So just very different project to give you an idea about where the microbiome can play, which is basically everywhere. I mean, that should be the key. Today is that bacteria are doing everything and anything. And if you're like, well, no, that probably doesn't have an effect there. Yes, it does. So that's the key.
Whoa.
Yeah, right.
So we are not scientists. But wow, it was so fascinating to hear everybody's introduction, because I feel like we probably have read papers that you've been involved in or research that you've been involved in. So it's really an honor to be sitting up here with you guys. Thank you for your work. Wow, it's really intense. So yeah, we make sauerkraut. So we make fermented foods using the power of fermentation, which is the oldest form of food preservation. So before there was refrigerators and high pressure canners, vacuum packed sealers, we as human beings subsisted on what we could store in cellars, what we could cure, and what we could keep through the winter. Over time, that sort of art form has kind of died off a little bit as we've become industrialized and fell in love with our refrigerators and microwaves and vacuum sealers and all these things that have kind of deadened food. And there's been a cultural shift to sort of revitalize this practice. And so we've sort of taken that into our fold a little bit for flavor.
For flavor, and then, kind of a systemic response to industrialized food. In a lot of ways, we feel, and a lot of people feel when they change their diets, how lacking live food is from most of the Western world. And interestingly, so much of your research kind of deals with the gut, and so many of the studies that we hear about are often about antibiotics and medicine. And so often I'm just like pounding the table like, food!
Where's the food?
There's so much dead food that people eat, and as biological units walking the earth, that's new to the last, just 50 years, and increasingly each decade, since. It's just this hyper fast deadening and just, removal of the biology of that, daily interaction.
Well, you made some very nice remarks about us, and let me pay some homage to you. For example, it's been known that we've had an obesity epidemic that's been going on for just really pretty recently. You can look at the numbers, and epidemiologists will tell you how much we've really increased. And there are ways in which you can blame it on food, but there are, for example, there's a lot of tantalizing evidence that it may be associated with the increased level of antibiotics. Okay, and so just to be clear, the potential relationship is that the gut that are storming away, in the bacteria that are in your gut just storing away on this food, they are essentially a recycling plant that help you sort of convert over as many different nutrients and as many different things as possible. If you are exposed to antibiotics, you're going to change the overall diversity of the bacteria in your gut, and you're basically going to reduce its recycling plant power. And then it presents your body potentially with more things like fats that will have a potential role in increasing your obesity. There's not necessarily a direct relationship between this, but if you look at a plot of the United States and you look at the states that prescribe more antibiotics, they are directly related to the states that have a worse obesity problem. Okay? So those of us who are thinking about making our food more of a living entity, I think there's a lot of different people that are talking about probiotics and a lot of different information out there that really isn't scientifically based. But thinking about using natural fermentation and the sort of the bacteria that are in this environment right now, we can call endogenous bacteria. And there is very definitely a hypothesis that is floating around out there that the more that our children are exposed to endogenous bacteria, the more that we're protected later on. We know that with breastfeeding, there's greater protection, and that is a direct result of receiving your mother's microbiome. We know that children that grow up in rural areas are less susceptible to cancer when they're older, because probably the immunological challenge that they're presented with bacteria. So I am all for and pay homage to those people that are thinking about introducing more living food in our diets, and I love kimchi.
Alright. That's where you should hold up a jar of Hex for me.
Why you didn't bring any is not clear to me. I will take some home.
I have to tell you, I live in Guildford, and so there's all these Hopkins students, right? And they're constantly sold out of Hex. Constantly. They're all filling up their little baskets along with their hot dogs and their white bread.
Recommended by
Well, at least they get one thing right.
science.
But you're right, that is something that's really been lost over the generations. I mean,
There
as
is
we've kind of
this
switched.
one study that this small village valley in Bulgaria somewhere where all these people live to like 100, 120. And then at some point, the researcher went in there because they thought that there was something unique in their genome that, these people lived forever with no cancer, no chronic disease, no nothing. And they sequenced a bunch of people and didn't find anything. And then at some point, someone was realized that they had these fermented, milk product, that yogurt, that they would put everywhere, like the commercial, I'd put that stuff everywhere, yeah. So it was, and the funny thing is that, so they had that ferment, that specific, so it's not a unique bacteria, I think it's a mix of different bacteria that they had used for literally centuries, so that they would propagate, and basically, that thing they have been using for hundreds and hundreds of years, and then they realized that this ferment had properties that were, anti-cancer, anti-inflammatory, anti-everything, but they literally eat, kilos and kilos of it every single day. So, they're on a high dose. But just to say that it's not, it wasn't their physiology that allowed them to live basically disease-free until, 110, 120. It was just the stuff that they were putting in their food.
But or is it though? Because you're a physical human being in that environment, and you're isolated in a sense, and you're eating all the food from that environment.
Right.
And it's cultured.
But look at the studies on probiotics, for example, in preterm birth. So there's 5 random control trials out there. 2 when they started probiotic supplementation, the first trimester, significantly longer gestation. Other studies on probiotics, and to be honest, I'm going to ask these guys, your food is really more prebiotic because of the fiber in it.
Yeah.
Symbiotic.
But that's actually
Right,
the
that's-
really one.
Right.
They have a lot of
Yeah.
live bacteria in their food as well. I mean, and because that's what,
Yeah,
I mean, can
because-
y'all explain the fermentation process and how you actually go through it with whatever particular product you'd like to talk about? Because I just remember being amazed that you're really just cutting up and squishing up cabbage and it doing it on its
That's
own.
basically it.
And
But-
then, like given
but-
the specificity of all of this, it's important to kind of remember the larger cycles at play. Like decomposition and recycling. Not just in our guts, but on the planet as a whole. And so we just kind of excerpt one little aspect of that and manipulate this really particular environment to our benefit to be able to store foods for later use, which is very functional and utilitarian in its early invention. How to eat through times of scarcity, and then it turns out when you pull it apart, there's a lot of wisdom in that survival, and then it is multifaceted, specific to our survival, but
Do we know
specific to
when
that.
it started, really? Does anybody have a good?
As soon as someone stuffed some
Yeah,
excess,
there's no.
food for later consumption into a pot or a goat's stomach or into a pit, or every culture has a fermented food, that was born out of, how are we going to eat this when, the yaks
That's
are-
how you make alcohol as well. So,
One of those ideas that really fascinates me is about alcohol, like as the cerveza, was sort of like co-evolved with, our biology.
Oh,
This,
very much.
just a kind of a fascinating, mutual selection, but specific to our process.
You take vegetables and you chop them finely, and you add preferably a high quality sea salt that doesn't have any other things added to it, so hand harvested sea salt.
And hopefully the vegetables are not radiated, which is
not radiated.
common. A lot of dead vegetables that come from elsewhere, and when they come in, they preserve longer when they don't have these decompositional microbes on
Right.
it.
Yeah. Salt naturally extracts water. So you have the water that's extracted from the vegetables. You create an anaerobic environment, so there's no air. And you cap it off and you let it sit. And you let it slow cook for months in its own microbial juices.
And as
That's
it
it.
sits, it acidifies and goes through all these transitions, and fascinating sort of transition from different stage bacteria. You can eat a young ferment and have a different composition of bacteria than if you ate a late stage ferment. There's hybridized ferments where you have sort of like that notion of symbiotic. You have the fiber that's feeding all the furthest reaches of your gut, all those bacteria, and then you have sort of early stage bacteria. But that's sort of where it gets super sciencey, and where we're just short of like plucking the wings off a butterfly to see how it works. We love the magic of it, and want to concentrate a lot on connecting with farmers, rebuilding food sheds. A lot of our food very intentionally was interrupted to come from afar, and there's a lot of policy in place that we're trying to reestablish to connect farmers with local markets and, have a viable food business.
Well, I guess the other blow for sensibility that I would like to applaud is, I was recently at a microbiome meeting that was specifically on metabolism, diabetes, and obesity. And the connection now between the traditional Western diet, which is high fat and a lot of carbs, and how it's impacting our predisposition to diabetes and obesity it's just there. It's a no-brainer, and they've demonstrated what's called causality. It's easy to show that this is causing this problem, and there's a very large role of the microbiome. So, as an example, you can take mice that are fed on a high-fat diet. They become obese. And then if you just simply put the mice that are lean in the same cage, they'll be exposed through the pellets that are the fecal matter that's in the cage. They'll be exposed enough so they will gain weight. So there's an obvious change in their microbiome, and there's an obvious change, and that is very definitely happening in the human population. And it's the high fat and high carb that's sort of the smoking gun that's definitely associated with this. And then people brought in another axis of how much this then is very closely related to a higher incidence of cancer. Okay, so anybody who's doing anything to change our dietary habits to be eating high fat and carbs and moving that over to the spectrum of eating vegetables and essentially pre-digesting them for us.
Yeah.
So they're even more accessible. Again, I think that this is a very, very powerful lifestyle change that I would certainly like to see in terms of our population and where it's going.
But we have a really significant problem here, because I can go to Eddie's and afford your product, right? But the patients in our preterm birth study, right,
Yeah.
they don't eat fruit and vegetables because WIC supplementation doesn't really give them very much. And so most of the WIC supplementation is actually quite high fat. And that's not to knock a supplementation program. It's a fabulous program. But they go to McDonald's because for $2 they can feed themselves. So, here we are in the richest country in the world, and we have the poorest nutrition, poorer than nutrition in Africa where I grew up. So, I don't know how we,
But along those lines, I'm sorry, just right quick, I think but y'all are big on workshops and teaching people. My mother grew up, really poor in the hills of Tennessee, and she was telling me about when I went to y'all's workshop and came back and was like, "Oh, sauerkraut, this is so awesome, we're going to do this." She was like, "Well, we always did that. This is the only way you make your cabbage, work through the winter, and beans, that's all we ate because that's what we could afford." But there's that knowledge missing, because of course she was generations of farmers in the hills of Tennessee. So she had other people teaching her, how to grow the cabbage, how to chop the cabbage, and we don't have that in
it's
inner
more
city
than knowledge.
Baltimore.
Because most of the women in our study know. I mean, we do
Okay.
diet tracking the whole way through pregnancy, basically. And we give them cell phones, and they take photos of the food, and then we analyze the content. And it's kind of funny, actually. We said to one the other day, we worry about portions, right? So I said to one woman the other day, okay, so how do you decide portions? So she said, well, I always photograph the one food that I eat. But if I eat the same food again in the same meal, I don't photograph it. Well, what do you mean? She said, so if I have a second sandwich, I don't take a photo of it. Okay. All right, we got it. But, so we watch their diet the whole way through. So we do find that the obesity issue, it's more than just fat. It's to do with the drinks, right? So when you get a 2-gallon tub of Hawaiian punch, I guess it expedites what happens. I don't know what happens to the food, but something happens. So the BMI in those women go up much higher, even though the calorie intake is lower.
I think that the response to liquid, especially liquid sugar versus solid sugar is different. So I think that
So it's an insulin.
so I think it may
Yeah.
be, yeah, somewhere maybe potentially through the gut-brain axis or something. But I think another point I just wanted to make about Owen's comment was, within our own generation, consuming a poor diet, low in fiber and potentially low in they call it microbiota, accessible carbohydrates. This group out in California and Stanford just recently showed a really interesting finding that intergenerationally, we're seeing extinctions of entire populations in our gut, potentially in other niches also, that when you consume a low fiber diet, you maybe lose 30 or 40% of your bacterial diversity. Then in the next generation of your offspring, they're inheriting those microbes. And even if they were to consume a high fiber diet to try to reconstitute what their ancestors had with their diversity, they can't quite achieve that unless you reintroduce some of those ancestral microbes. And so I think that we're also interested in what are the ancestral microbes that we've lost. That's why a lot of groups are looking at the Hadza warriors, the Yanomami down in Venezuela, the hunter-gatherers, the agrarian populations, and finding out where we were and just kind of profiling that. Because I think in the future that could potentially be a big game changer. Because we might not be able to achieve what we once had just through a high fiber diet. And I think that's where maybe probiotics could come in the future too.
Yeah.
But I
Or
think there's-
eating living food.
For sure. And I think that there's certainly a need to inform the public, give the public information that they can rely on too. That's why I think there is a need for good science also around what are these doing, can they actually improve health, or is it just a claim that's being made? But I think that there's so much potential that it would be great to work with more food scientists and people like yourselves who can kind of rigorously test these things.
So much of it sounds like an issue of balance, and just hearing you say that reminds me of a study we read about H. pylori, how you've been indicted for causing stomach ulcers.
Right.
And it was sort of, let's eradicate this,
Exactly.
H. pylori, get out of there.
Yeah.
And then it turns out, like most things, in balance, with it in the right communities, it has a vital function in the body, but yeah, balance seems to be such a big aspect of, both all of your research, and I think.
Well, I mean, one of the things on that subject, I like to play a role in sort of reflecting back stuff, the scientific basis of stuff. So like, when you make a comment like that. So one of the things that I find very, very satisfying about what we're doing here is for a long time in our field, in any respective field that was coming at microbiology or molecular biology, just trying to understand, we really, really, really almost always were rewarded for the science that like pulled systems apart. Like it was very much this isolation system. If we wanted to understand an enzyme, we would just look at a specific enzyme. We wouldn't look at it in the context of what was happening in a cell. If we wanted to understand what a bacterium was doing, Emmanuel described very, very well how you actually started just looking at a single bacterium, even though in your heart of hearts you gosh darn well there was plenty of other bacteria in there, and we weren't characterizing them at all. And one of the things that's really exciting about the microbiome work is we actually get to look at a holistic system and start to ask rigorous scientific questions about it. And there's something very gratifying about that, because we all know in our heart of hearts that we're talking about a full-blown ecosystem that's got lots and lots of dynamics. And now we're actually able to do that. And so you mentioned, you used the word like balance. Heck yeah, not only do I want to know about balance, I want to know about the whole system. And it's incredibly dynamic, right? I mean, there's times when a whole change could sweep through, essentially like a wildfire for any of these systems, but they grow back, and we're just beginning to understand that. There are temporal studies about different times of the day that you could be looking at your microbiome in your gut and see differences. And so there's cycles that are happening at that level. And then if you look at things like the Chesapeake or ocean systems, You can see cycles that happen on a yearly level. And so it's a very, very exciting time for us. And it's a very gratifying time in that we're starting to think about things in terms of an ecology that's really new.
I just like that thought of the rainforest, the indicator species,
Yeah.
and how they all interrelate and keystone species, and
Yeah.
one goes missing or is underpopulated, and then a whole cascade of stuff that happens.
Right. Yeah.
I guess it's part of y'all's work to find out that the balance, like what's a healthy snapshot?
Well, I mean, so one way I've been thinking about that is that, like, there are some species that are actually in incredibly low abundance and they control everything. If you go to Yellowstone, the thing that sort of impacts virtually everything, including the population of trees, et cetera, is the wolf. Okay, they're the guy at the top of the food chain that are really influencing the sort of species all the way down to the level of bacteria, lots of other macroorganisms, the squirrel population, etc. And yet, if you took a block of Yellowstone, like if you took some volume of Yellowstone and sort of did a survey at a genomic level of the total amount of wolf that you find in there, it would be much, much smaller than everything else, than the prairie grass or the bacteria or the whatever, yet they're kind of controlling everything. And I'm told that in ocean settings, like the otter does exactly the same thing. So it's the predators are at the top that sort of is actually impacting everything. And we don't appreciate that about the microbiome. We're always looking for the most abundant species. It's kind of hilarious, right? But we're not actually looking for, who knows if they're predators, but for example, we do know that there are viruses that are also dramatically, we'd probably be one big bacterium if it wasn't for viruses in our gut that are also there, and they're kind of keeping the overall population at bay. So that's another dynamic of this that we're just beginning to understand.
Right, I was actually going to mention, I imagine if we had a virologist, especially a phage
Yeah, that would've been a great
they would
idea.
be arguing that
Yeah.
the phages are taking care that they're, exactly.
So
Yeah.
true. They're the ones controlling the system, right?
Right.
And there's a legit argument for it.
Yeah.
Right.
It's ironic that we've known about ecosystems for millennia. But if you take this out, it's going to affect the whole ecosystem. And yet our approach to health has always been not to leverage human physiology, but to obliterate it. So we're going to give you antibiotics. We're just going to do this
Yeah.
with anybody who has surgery here now. You have to have one dose of antibiotics before they open you. One dose. That's like the worst thing in the world you could do for bacteria, but that's what we do routinely. Practices like vaginal exams in labor.
Yeah.
Labor is a perfectly normal, healthy thing to do, and yet 98% of women have epidurals in urban settings. In one data set that we have now, over 90% of them had rupture of membranes, and then had oxytocin to augment the labor because they only had 24 hours before they went into it. So, you've got, this is healthcare now. So the microbiome offers us this opportunity to step back and say, hold on. So for example, at the NIH, they're researching certain bacteria that they can use in place of antibiotics. So we give you one bacteria to treat another. I was in a presentation a few weeks ago, and a colleague of mine was telling me about a grant that they did. They identified an antibody, and this antibody lyses HIV. On top of that, it also renders the woman infertile. And so, there's this fabulous thing that we can do. We can, give it to a woman and it protects them against HIV, but can we turn it off? So it's what you say. We change one thing, and we may introduce a whole other set of problems. So I think for most of us, it's about stepping back and saying, okay, let's understand these systems, and then let's leverage them. But, even you'll be old before we start that.
Well, so
Clinical colleagues don't necessarily disagree. They're like, oh, is this going to be another fad? I'm like, no, it's much different.
Well, and that's what I wanted y'all to talk about was, when do you think medicine's going to really embrace this? Because there are some aspects of medicine that are a little bit, but I've heard so much more backlash. Because one of the things I write about a lot is pregnancy and birth and our first foods and things like that. And a lot of the backlash is more from the medical community than it is, you know, the midwives are pushing a lot of this stuff and supporting it.
So there's, I'm sure everyone here could talk about the different obstacles to the process. So just to give you an idea of one, if somebody came up with sort of a slam dunk probiotic, okay? Part of the issue is that right now there are some things that have been approved by the FDA, the attenuated bacteria that are prescribed specifically to protect us from different bad bacteria that are out there. So that you basically inject or eat these bacteria and you develop immunity to them, and then they're sort of lame bacteria. They don't cause any disease, and then you develop immunity to them. In order for that process to go through, you have to sequence the entire genome, and you have to characterize every protein as best you can in that genome, and then the FDA is willing to classify it as like something that's, okay, this is FDA approved, et cetera. Okay, now let's cut over to microbiome. These guys grow something off of kimchi. We discovered that it's a slam dunk. You could always lose weight with this thing, okay? Well, that's a collection of bacteria, okay? There's like so many different species in there, you can't even begin to, it's like assessing this block of Yellowstone and saying, tell me about all the organisms in there. Then you'd have to sequence them all. Then you have to characterize them all. And the FDA, I've been to the meetings where they've done this. The FDA have gotten together the scientists, and they've got together people from pharmaceuticals, et cetera. And the FDA wants to approve these. They're not obstacles. I mean, they genuinely want to approve these, but they've got their own process for doing it. So that's just sort of a one example. But there's actually a lot of hope. I mean, there are certainly clinical trials going on for probiotics, et cetera. And there is some hope that it may start appearing. But for example, the C-sections, like if you could argue that it's healthier to pass down the birth canal versus C-sections, you tell me, when would that actually turn into routine standard of care?
Exactly.
You know,
You probably worked for it,
So
actually.
yeah, so we're working through that with IRB issues, right? Because they say, oh, you're introducing bacteria that could potentially be pathogenic,
Right.
and give these, potential risk. And so Maria Gloria published an observational study where she swabbed a couple of babies, compared them to C-section delivered babies and vaginal delivered babies, but it was all observational.
Yeah.
There was a BMJ editorial from clinicians who said everybody's asking for the seeding procedure, and like we have to tell them that there's all these potential health risks, and they pretty much just said this is a bunch of rubbish.
Right.
And so our argument is that these are the microbes that babies are naturally receiving, and clearly any indication that we wouldn't allow for a vaginal delivery, we wouldn't allow them to have a seeding procedure. Of course, there's interactions with other things that are going on during vaginal delivery, that we can't necessarily counteract in the microbes that are conferred to the baby at birth. But I think there's a good argument to be made that those were supposed to be inherited from at birth.
But on the subject of how long, agonizingly long this takes until we change things, like, so you just described an IRB, which is the review board, the internal review board at Johns Hopkins that you have to kind of convince. Let me just do a research study, okay? A research study is different than a full-blown clinical trial where you've got hundreds and hundreds of people that you enroll and show that it works. And then once you've done that, you have other steps that have to go through in order for it to become kind of a new standard of care. You probably have more, I'm an engineer. You probably have more, but the point that we're trying to make is it's an agonizingly long process, even when it's non-adversarial. I mean, even when people aren't really against it, it takes a long time to actually change the standard of care.
But I don't think it's reasonable to expect clinicians who grew up and were trained in an environment where bacteria was the lethal foe.
Exactly.
Yeah.
To
Right.
suddenly switch
Right.
and say, whoa, come on in, E. coli.
Yeah.
It's not and the other thing is, and particularly for nurses, like we're on the front lines, and those nurses are held accountable for patient infections. So, it's tough for them. It's really tough. But the seeding thing. So there is a Brazilian researcher who's in New York now, who has, she puts vaginal packing in women who are having planned sections. And then when the baby's born, she actually wraps
Maria
them.
Gloria?
Yeah.
No.
Yeah.
Is that?
Yeah,
is
you're
that
talking about Maria Gloria.
Yeah.
Yes.
So, and the findings have been
Yeah.
incredible.
Yep. Yeah.
Yeah.
So you have women out there now, mainly social class 1 and 2, who read all this stuff and
Right.
buy the New York Times,
Yeah.
right? Who now are asking, well, can I get some packing? I'll do it myself. And physicians don't know what to do.
But I would say that, we have to be careful because
So
the
the big
incredible results are like a little bit has been
And
stored.
also-
We have no idea
And
about
so
how.
basically
how
just rub the
well
baby
it's
with it.
That's
Because the
But
baby
it's has
will
potential.
naturally be exposed to it during birth.
Hey.
So if you have a C-section.
What's
then
going on?
there's no-
We're talking.
So
So those of you who know-
the baby is swabbed and all
Yeah.
the vaginal secretions of the mother.
Yes.
Okay.
Yeah.
Yeah, because the idea is that naturally when a baby comes down the vaginal canal, labor is such a long process that, the baby is actually swallowing vaginal secretions as it's coming down the canal, as well as being covered. Of course, there's, nowadays everybody scrubs the daylights out of a small human once it comes out of you too. So no one's really looked at that I know
Well,
of.
so there is
But-
some work on that.
Okay. Oh, great. You'll have to tell me that.
So actually the protocol is that the baby is delivered onto the mother's tummy and covered and not touched for an hour.
Right.
So how we're moving now towards that is to bring in the baby friendly, because baby friendly requires that the baby is not removed from the mother. But, the problem is large portions, particularly of the low socioeconomic group, don't want to breastfeed.
Yes.
So, and they don't want to breastfeed, and, I always kind of surprised me, but doing this one study, these women explained to me there's no room for them to breastfeed, even though this is federally mandated, so there's no room for them to breastfeed, they're not allowed to take breaks to go and pump. They can't take their pump to work because they don't have lockers and they're not allowed to put the milk in the fridge. So how can we promote breastfeeding in an environment that supports that?
Reminds me of another little thing we read about, that as yet unknown protein, I believe, that was in breast milk, or something that was unknown. It was like this
Enzyme.
enzyme.
Outside of the cell wall.
Yeah, but
I just wrote about it. That's the only reason I know
yeah,
that.
which I thought was just fascinating,
Yeah.
and kind of this, sort of aha beauty of,
Yeah.
biology, but that it, feeds and helps to populate the baby's gut and microbiome.
Yeah, so this is
Just
worked out of
fascinating. Yeah, of
Yeah.
course, breast milk feeds the baby, and
Well, if we were supposed to drink cow's milk, then we would give milk to cows. But it's very hard to get that message over to women, and there is this perception of body image for women. So when I was doing my dissertation, we had this really odd thing that we couldn't explain, and there's a sociologist in Canada called Lorraine Code, and she called it the sacred space, that somehow the vagina, this last 4 inches of birth, has become the sacred space. And a lot of it's come out of people like, what's her name, Beckham, who choose to have sections at 36 weeks because they don't want to gain the pad of fat that you get in the last 4 weeks, and who promote this idea of surgery, major abdominal surgery, to maintain this kind of feminine view of the woman, which was quite astounding to me. And I didn't do a lot of sociology, but that kind of really appealed to me because it made logical sense. But now with the microbiota, and with one of the data sets that we're looking at right now is labor data sets. And so the lubricants that we use to examine women during labor have antimicrobial effects in them. And so we watch the lactobacillus, literally. The lactobacillus colonies are wiped out
Wow.
at the end. And one of the patients we were looking at today, because we're doing like longitudinal, and rather than putting in a model, we're looking at them individually right now. And this woman had vaginal exams from 24 weeks onwards every month. And so you would see this slight resurgence. Primarily Barbarella grew and grew and grew and grew. But what was really interesting was that the pathogens would rebound faster, and they would actually double. and triple. And then as she went into labor, her read count of Gardnerella had gone from 2,000 to 20,000 in 20 weeks. So, yeah.
Well, I wanted to have some time too for y'all to ask questions as well. Does anyone have any particular things they'd like to ask?
They're probably all terrified now.
I was just, this is a question for the carboys. I was wondering what makes your fermentation process or your business in general different from your competitors?
There's no competition. Yeah. It sort of goes outside of the micro.
That's all right.
Well,
Yeah.
we work really hard to support our local farming community. So, 95% of the produce that we ferment is grown within the state of Maryland.
Grown organically.
And it's grown organically. So we don't choose farms that use pesticides or any sort of chemical treatment. They also use fair labor practices. So they're hiring within the community and making sure that the people that they hire are getting paid a fair wage and have good working conditions. So that's one. Two is we play with local flavor. So to that end, we're not, there are things that we outsource organically, but we try really hard to use herbs and spices that are grown within this region to come up with unique flavors. So that's another aspect.
Yeah, I suppose flavor is local, but sort of culturally wide-reaching in a lot of ways and play with a lot of ideas and recipes and interpret that in a way that's uniquely our own. So, for example, we keep a nuka pot, which is a Japanese style of pickling and rice bran. And so we make sukemono from things that we bury in this pot. Whether it's daikon or garlic or whatever, but it comes out as just, what is this flavor? And so using some of those flavors that we'll incorporate into some of our small batch ferments, the flavor, the farms, and then the health considerations, I think, are the kind of trifecta of what makes what we do, our approach, especially unique. There's a lot of pale comparisons, and then sort of, this is, I think, a lot of the cost issues you mentioned in McDonald's, and I think that food policy discussion, could take up another 3 hours. But, like, what does food cost and what we're up against, and what do we do with, sort of, and how do we treat, the people that work for us, our community. Right now we're a very small business, but we give 5% of our profits, at least the last 2 years, have been to Great Kids Farm. They go back into the Baltimore City Public Schools. The rest of it all just goes into buying equipment. Who knew a walk-in cost $25,000? So we we didn't know. We were like, what? We're going to a different company. And then like the next company was like 26. Like, what? It's a walk-in, like these walls that you like knit together with 5 screws and an air conditioner cost.
We also don't ferment in plastic. So a lot of major fermentation companies and minor fermentation companies ferment in U-line plastic barrels. And we always have thought, even as, myself growing up, my grandmother made sauerkraut in the crock in the basement. Our parents canned and pickled. Why would we take something and why would we put it in plastic when it acidifies and eventually will eat away at that plastic?
Yeah.
So we've spent a lot of our business dollars into buying high-grade stainless steel fermentation vessels. So we use very high-quality equipment, which we in turn know that it affects the end flavor
we could buy barrels for like a
$45.
20th of the price of like a variable capacity stainless steel tank. But now there's bacteria used in bioremediation, you know, to like break down plastics. But you'll bet that that stuff shipped from California that sits on the shelf for a year in plastic that was subsidized, you know, to get here and grown then under a drought. You know, it's just, there's so much screwy stuff that happens with food, specifically with, you know, even sauerkraut. But when you look at it on the shelf, you know, it's like, oh, it's cabbage and salt. And, you know, like, what kind of salt is it? Is it hand-harvested Celtic sea salt, you know, with 32 other minerals that help, you know, inform our body and bodily function? Or is it sodium chloride? You know, like all those little things and, yeah. Well, yeah.
But there's not just two of you and your Japanese wok, right? I mean,
No. we employ almost 10 people.
Oh, wow.
I'm surprised it's only
Well,
Yeah.
five full-time and 2 part-time.
Well, no, we have a staff of 10 people, and it's
Well, including us, there's 9.
we have a staff of 10 people.
Okay, that's
Yeah, I mean, but like it takes, you know, it takes a lot of effort, you know, to do farmers markets and bulk food, or wholesale, and having a shop, and you know, maintaining a high quality of standard and cleanliness.
Yeah, I mean, that's the tough part. We don't sell iPads, you know, or like, you know, electronics, or, you know, it's tough. There's really low margins on food. And, you know, people are like, what, $10 for a jar of sauerkraut? And we're like, we're so small batch for so long that $10 was just like barely paying for it, which is ridiculous. But, you know, with the hope of building in these efficiencies to make it, you know, a viable business, and we're getting there. We just moved into a new production space, and that has been, you know, transformed. Well, just moved like last week, but it's already very transformative and potentially transformative.
Well, I was at the University of Indiana last week, and they had your product in the, they actually are renowned for the student eating place, whatever you call that, cafeteria. And and they had your product there.
The
Really?
University of Indiana?
Yeah.
Really?
Yeah.
Like a student run?
Same brand, huh?
Yeah, the Hex brand, yeah.
Oh,
Really?
far out.
Oh, okay, cool. That's awesome.
So you're getting out there.
I
Yeah.
was talking to somebody in Nashville that has kind of like a fermentation museum and a root cellar, and they want us to send them stuff. So that's kind of neat. Yeah, for something so base and accessible, you know.
Yeah.
And then, you know, I really feel like this is all part of a very cultural trend as we become more and more virtual and dissociated with our environment, and everything's so virtual. You know, touching things and, you know, being place-based is very important for our psyche and interrelationships and a lot of other things.
You have a question?
Yeah. So just hearing everybody talk about these things, you know, this whole project is all about complexity, right? It went from like, we have like this one disease, and maybe there's these factors like, oh, it's actually this whole ecology, and there's these thousands of living things that are changing over time, and they're making, you know, hundreds of thousands of proteins. Like, so, I mean, how do we address this? I mean, our brains haven't gotten that much bigger that we can, you know, hold all this stuff in our heads. You know, we're getting faster and faster computers, but that doesn't seem like that's necessarily going to solve it. Like, just more, you know, more bits, like, doesn't create the whole picture, doesn't tell us about the relationships. Is there any sort of strategies in terms of like systems thinking that we're sort of understanding these and being able to sort of unlock these things from a slightly higher level than like knowing every damn nucleotide and trying to solve at that level?
So I can take a shot at that. I mean, There's a number of different levels to answer your question. But one is that there are an awful lot of very, very elegant experiments going on where you could essentially treat the microbiome as a black box. You don't have to know anything about it, except, I can raise some mice on high-fat diets and raise some mice on low-fat diets, and these guys are lean, these guys are overweight, and if I put them in the same cage, then something changes. And that's a very elegant experiment, but you don't necessarily know what's going on, but it's enough to start, leading the way to other different things. So don't underestimate the value of, in a lot of ways, people in medicine really don't care what's happening under the hood. They just want to see a change, right? And so if you can start to sort of point to this works and this doesn't, if you change this simple aspect of standard of care for women during preterm birth, we get a higher success rate. They're like, okay, fine, but they don't need to know that much more about the system. That's one aspect of it. Another way to answer your question is math. I'm aware of a study right now that in order to follow diabetes, they're looking at on the order of 10,000 kids who aren't diabetic yet, but they're looking for the incidence of them becoming diabetic. So they're tracking all these kids and waiting for that changeover to happen. And they're collecting samples, let's say, on a weekly basis. That's an enormous amount of data. That's just, to give you an idea, these files, in order to open them and to have a computer just touch each nucleotide to count them, for example, and then close them, takes a half an hour. Okay, so it's a lot of data, and it takes a long time, and we have big grid computing systems. I have a 4,000 node computer that are used to apply against this stuff. But in order to really understand it, to comprehensively understand it, no kidding, like Newton invented calculus in order to understand some of the systems that he wanted to understand. And no kidding, there are a lot of really, really brilliant math people that are coming to the game, and we're detecting trends that you absolutely would not find if you looked at smaller numbers of people. So another way to answer the question is, I say it playfully, but there's really a new development of mathematics that's sort of being brought to bear against this to find interesting trends. And then the other thing is that my background's engineering. I'm much more of a brute force engineering kind of guy, and we are putting online really, really big computing systems. And I have a pilot study right now, for example, with Amazon. It's like, the idea is that if we stage a bunch of data up there and people can, essentially break out their credit card and rent the computing time in order to answer a question, that's the type of thing that we'd like to enable people to do. The NSF is traditionally the organization that has been funding things like the Hubble telescope and other activities like that. A lot of the terrestrial telescopes, what they do is they build these things, and then people write another NSF grant to just rent time for 24 hours to use that system. The NIH is basically looking at exactly that model, that they could partner up with a place like Amazon or someplace that's not necessarily a for-profit entity, and you'd write a grant and just put in the request for, you don't buy the computing equipment, you just put in a request for the computing time and run your analysis for X amount of time and get back results. So that's another way that we are starting to approach the problem. But you're absolutely right. There's a degree of complexity when you start to want to ask pretty straightforward questions, like tell me what happens in the population of kids that when they convert over to diabetic. You have to do these like the equivalent of these kind of moonshot kind of projects. They get very ambitious very quickly, and it's just an absurd amount of data.
Well, and by the same token, the more variables you have, the larger your sample size has to be.
Yeah.
So now, like on our grant, we collect oral, GI, vaginal, we collect from the neonate, we collect blood from the mother and the baby. And the NINS is a $3 million grant, right? That's just direct costs. All of that data is going to go into the NIH's databases, and any research can access it. So suddenly we've gone from an N of 300 to multiple studies, and you can have 15, 20, 30,000. Some of the health databases, you can have, like I have access to 3 million births a year, right, if I want to just look at birth record data. So we've got bigger data that can capitalize on that map.
So I have a million questions, but I'm only going to ask two. I have one specific and then one pretty big picture. So the one is certainly for both of you. So I've been reading a little bit about how they keep saying the sterile environment of the womb, right? Which is most likely not true. So do you know the data, you know, what kind of data is there? And I mean, it's probably pretty hard to test that often, but to figure out, you know, the diet of the mother, et cetera, all these things that are happening pre-term, you know, actually before they're even pregnant, and then when they're pregnant, to the microbes there. Yeah.
So we know that the whole uterus and tubes down out into the ovaries, right, is colonized with bacteria. And if you think about it, when fertilization takes place, right, it closes over.
Mm-hmm.
So you likely trap bacteria there. But there's some interesting work looking at mainly placental stuff. Some of the intrauterine, when they pull the sterile, they used to think was sterile liquid out, they've grown cultures. These kids come out colonized.
Right.
I mean, yes, they pick up some stuff through the vagina, but we know, for example, that a labor is initiated from within the fetus, right, by bacteria. So we know it's there. We just, how can you ethically study it?
Yeah, yeah.
One of the things we're doing on one of the studies is we take a groin swab from underneath the vernix when the baby's born. We're working with the guy at Hopkins, and we believe that actually will tell us what their colonization was in utero, because the vernix actually seals the bacteria. The problem is, going back to your issue, you know, the grants aren't big enough.
Right.
You know, $3 million covers the vaginal swabs of 62 women. That's it. You know, we would need $15 to $18 million to analyze all the data, and we will over time. And then, you know, you meet other researchers and they're like, well, I've got a little pot of money, so I'll do your oxidative stress assays for you. I'll look at the antibodies, right? So, you know, we all, I think, kind of do little bits of things, and ultimately the knowledge will grow.
Right.
But the bacterial, the intrauterine, and I'll tell you now, this just blew my mind. So a nurse practitioner, she over at she works with babies that have what are called short gut syndrome.
Oh,
So
yeah.
kids that have necrotizing enterocolitis, they just cut back the gut, and some of them end up with like literally 4 inches.
Yeah.
So they've started to colonize the blood, and they're finding gut microbes in the blood of these kids. So you know,
I was just at a meeting where there were one session specifically on bacteria in organs. So this was at a for-profit, but they were studying people that had a certain type of cirrhosis that was not cirrhosis associated with alcohol. Okay, so that's basically scarring in the liver, right? And they're not making any claim that bacteria are causing this. But they are making the claim that there are bacteria in the liver, and a different population of bacteria will grow in response to cirrhosis. And they can serve as a biomarker for cirrhosis. So, and you can pull them out of the blood and use them as a biomarker. And they were also studying this for Alzheimer's. So
autism.
where the idea is that there may be bacteria that serve as a biomarker for something that's going on in the brain.
Well, Stanford has a huge
Unreal.
program now for autism
Yeah.
and the microbiota.
Yeah.
So is there a single disease any of us can think of that isn't linked to the microbiota? A single disease process?
Go ahead.
Maybe.
I think it'd be challenging.
Oh, yeah, no.
Well, but let's be clear that there's a difference between, are there bacteria that are responsible for causing this, and are they coming along for the ride? So if you're diabetic, you have a completely, different type of glucose profile in your blood or whatever, you could imagine a very different profile of bacteria just as a result, not necessarily the causation. So we do have to be careful.
Well, and also that it's the bacteria produce metabolites that initiate proteins, cell signaling.
Yeah.
It's not like the microbes are going out there attacking the liver and giving you cirrhosis, right?
And I think also one thing to point out, just to cap off your question, is so that this placental microbiome got a lot of press, and the microbiome and the 16S sequencing at least, you can only really identify that there's some bacterial DNA present.
That's right.
But we
I
don't
see.
know whether it's
They're not
live
live.
or it's just like
That's an
phagocytic
important-
remnant, right?
But we can test
And
it.
I don't know if anybody's cultured that placental bacteria.
They
I think in the
Say-
paper they
they-
said that they tried, but they couldn't go any
Yeah.
They say they've got some fish data too, fish is fluorescent in situ
Oh, that was-
hybridization,
yeah, that
it's
was
just
just
where they've
full
colored
paper.
it, but I don't
But
know, it
fish
seems
is
like
really
the
safe.
controls aren't, the
Yeah.
controls could be stronger.
Yeah.
And I think with the
So-
placenta, we're moving more now towards epigenetics.
Yeah.
Because it
There's
is
a
so
placenta
difficult
project.
to study. The placenta's an organ, it's its
Right,
own
yeah.
organ. So I think the epigenetics movement will probably tell us more.
There's a question.
Yeah, just following up on what you said, I was very interested about, you said patients that have diabetes, that they would have a whole different type of bacteria that would be
Potentially, yeah.
different diseases as well. So in terms of biomarkers, is the scientific community, the research community looking at kind of like characterizing that and looking for like DNA fingerprinting, looking for a slew of bacteria as a method of diagnosis, like Alzheimer's
So,
or diabetes?
yeah, so I'm no expert, but my guess is that your standard issue doctor who's looking for diabetes would say, there's plenty, we just look at how you respond to glucose, and we know whether or not you're diabetic.
Yeah.
So what they would
Other
say,
diseases.
I think what they would say is, we've already got a 50-cent assay for
Yeah.
doing that works really well. But the potential is that there might be some bacteria that might be diagnostic of being pre-diabetic. They could tell you that it's coming, buddy, you should change your lifestyle. Meanwhile, there's plenty of other, as soon as somebody starts gaining weight, you can tell them, if you're not careful. But, that's what might be an interesting dimension of your question, is to ask if there are some bacteria that seem to be, sort of, can tell you about that you're in early stages, could they serve as a biomarker for that type of thing?
There is a study with colon cancer where they found really strong biomarkers with colon cancer now.
Right.
So that one's probably the best
And that's a
example
good
I know
example of you don't have early detection.
Right, you
And it would be
don't.
wonderful if you
Right,
did.
right,
Yeah.
because it makes a huge,
So was that like multiple bacteria or they looked
I don't
at
remember.
specific?
I really don't remember.
But that's something that, the field is moving to. So I have a project like currently trying to do exactly that for appendicitis. So, because
Oh,
the problem with appendicitis is that I think 30–40% of the time a kid or an adult will present with the appendicitis symptoms. They do an MRI, so massive dose of radiation, and then it turns out that it's just a bad bellyache. So what we are trying to see is if we could just take a rectal swab that would be non-invasive, and then just do sequencing and find and see if we can find biomarkers, so bacterial biomarkers of appendicitis. That would allow us to have a better diagnostic tool. We even patented it and everything. So